Okay, well, why don't we get going? I'll give a little introduction to Dr. Bailey, and then we'll look forward to his presentation. So Dr. Bailey is a professor and chair of child and adolescent psychiatry at UBC. His research has investigated the neurobiological basis of autistic disorders, and he uses genetic, neuropathological, and neuroimaging approaches. His clinical work focuses on teenagers and able adults with autism spectrum disorder. So his presentation today is the neuroscience of autism, and thank you so much, Dr. Bailey, for presenting.
Okay, can everybody hear me okay?
Yeah, I think so.
Good. So I don't want any false expectations. By the end of this talk, you'll know more about what we don't know about the neuroscience than what we do know. Now, how do I make my slides advance?
Just with the arrow keys, I think, go to
Okay.
Yeah.
So historically, people back in the 60s and 70s tended to think of autism as a highly localized disorder. So the thinking was that either there was an abnormality in the social system in the brain or an abnormality in the systems that subserved language. So if you go back to the bulk of psychological literature in the 60s and 70s, there was at times a fairly vicious dispute between these two groups as to which was the primary abnormality. And what changed people's thinking was the realization in the 70s that autism usually had a strong genetic predisposition, that most individuals didn't have single gene causes, and that one could see in the families of people with ASD individuals with milder but obviously related phenotypes. So people slowly but surely change from the localizing account to a model more similar to the sort that I've displayed here, which in fact is a complex disease model that was first erected for coronary artery disease. So the thinking is that individuals with ASD inherit susceptibility genes, and by that I mean genes that increase the risk of developing ASD but don't have a one-to-one relationship with the disorder. And those genes and any possible interacting environmental influences interfere with a neurobiological substrate. And the reason for stressing what seems obvious is that occasionally people refer to genes for theory of mind or genes for language, but that's a misleading shortcut, because these are not genes that are directly influencing behaviors which are at the top of the hierarchy. The model also has within it the property of coherence. The idea here is that if you disturb what would be the normal equilibrium at any of these stages in the hierarchy, then the body, or in our case the brain, will adjust to try and bring the situation back to a normal state. So in a disease state, we're not only seeing the direct neurobiological consequences of genetic influences and possibly gene-environmental interactions, but also anything that the brain does to get around those difficulties. Obviously, we're thinking about developmental disorders, and so we're not really having a static hierarchy in the way we have portrayed here. these factors may change over time. Obviously, genetic risk doesn't change over time, but neurobiological differences between people with ASD and the rest of the population may change, and clinicians who see adults with ASD are very used to seeing quite remarkable gains in adult life, which may entirely be due to experience-based improvement in cognitive functioning, but there's no reason to suppose that there are not neurobiological changes over time. And finally, a key characteristic of this way of thinking about complex disease is the property of emergence. And that refers to the fact that if you know the inputs in the bottom box, the gene and environmental influences, that doesn't mean that you can predict what the contents of subsequent stages in the hierarchy are going to be. And so as disease becomes more severe in whatever way we measure severity, new behaviors emerge. So in the case of ASD, we might think of epilepsy as being an emergent phenomenon, or mental retardation being an emergent phenomena, or early language loss being emergent phenomena. We don't know why some individuals on the spectrum develop these more severe manifestations of disease. So I'm quickly going to go through some particular aspects of that conceptual hierarchy, pointing out how people have used findings in these areas to come up with explanatory models and what the obstacles are really to moving forward at the moment. So let's start with the genetics of autism. Originally, the idea that autism might have a genetic basis was discounted because the recurrence risks seemed to be extremely small. But then individuals realized that although the absolute recurrence risk was small back in the 60s and 70s, it was high compared to the population base rate. And a series of twin studies in the beginning in the 1970s showed that there was a huge difference between the concordance rate for autism in identical twins versus non-identical. There's been one recent study from Hallmeyer's group which comes up with quite different findings, and Johan has suggested that this undermines the evidence for a genetic basis for autism. But in fact, if you look at the paper carefully, there's a fundamental flaw in that the twin register contains some 400 twins with autism, but the researchers only managed to assess just over 100 of those. There tends to be a well-known bias for parents of concordant twins participating in research studies. So the twin data still seems to indicate there's a strong genetic risk, and nowadays people usually cite a minimum recurrence risk of 5%, but the reality with the broadening of the autistic spectrum is this may well be 10% or possibly slightly higher. What's particularly interesting, and really an obstacle to very simple models of autism, is that in some other family members one can see milder difficulties that are obviously related to autism. So what this implies is that the autistic spectrum extends well beyond the diagnosable condition. And I think most of you are probably aware that the increase in prevalence of autism over the last 10 or 15 years is at least in part due to better recognition of able and less severely affected individuals. There are some single gene causes of autism, so tuberous sclerosis, Fragile X syndrome are the most common. But most people with autism, we believe, have the disease because they inherit a particular combination of susceptibility genes, but that inheriting the genes by themselves doesn't seem to be enough to cause the disorder, and we don't know whether the variation between whether you're affected or not and how severely you're affected is simply due to chance factors during development, or whether there are indeed genuine gene-environment interactions. And gene-environment interactions are important because when people think about the environment in autism, they tend to be focusing on environmental factors that are known to be intrinsically noxious: alcohol, cigarette smoke, lead, things we would all try to avoid during pregnancy or before. But if you have susceptibility genes, it's possible that environmental factors that in their own right may not be toxic or noxious may be detrimental to you if you carry a particular risk genotype. So over the last 15 years, a lot of time and money has gone into trying to identify specific genetic risk factors for autism. And the outcome of all of that is that quite a large number of genes have been implicated, but none of the genes by themselves accounts for usually more than 1% of the genetic variance in the risk to autism. In other words, we're still missing the bulk of the genetic effect. Now, people have taken solace from the fact that many of the genes have very well recognized functions in the brain, and therefore people have argued from gene to neurobiological mechanism. So there's been a particular interest in the fact that some of the genes are involved in synapse formation and migration. Some of the identified genetic variants are involved in cell migration. Some are involved in where axons that grow out from developing neurons go to in the brain. And we know that ASD is associated with, on average, increased brain size. Sorry, trying to suppress a sneeze here. And one of the identified genes, called P10, is known to have a direct influence on brain size. Now, this is very encouraging, but really these are quite different neurobiological processes, so it's not that all identified genetic variants are related to just one of these processes. But also, during development, many genes have what are called pleiotropic effects, and by that I mean they influence really quite diverse biological processes. So there are some genes involved in synapse formation that are also relevant to axonal pathfinding. So sometimes identifying a genetic risk factor isn't as much of a clue as to what might be the key neurobiological abnormalities you might imagine. But, of course, that doesn't stop people shouting from the rooftops about various things they have found. So this was a paper in Science back in 2007 that really kicked off the idea that autism might be a synaptic disorder. And one of the unintended consequences of the publicity surrounding synapse involvement, or possible synapse involvement, was this catalyzed the involvement of the pharmaceutical industry in thinking about getting involved in autism research, which was something they'd not previously been interested in. And the reason for that is that many drugs that have psychoactive effects operate at the synapse, and this is just a schematic showing some of the proteins that are involved. And in terms of mutations down at the bottom of the screen, people have identified changes in this protein, shank, in the binding over here of neurexin and neuroglians, and RAB3 is involved in the pathogenesis of tuberous sclerosis, as are some of the other proteins in this slide. One of the problems with postulating a synaptic model of autism is it's actually very hard to test that model directly in humans because none of our imaging approaches have that level of resolution. We can use radioligands in PET studies to investigate the density and distribution of receptors at synapses, but those studies involve administration of radioactive ligands, and they're not something that have been commonly done in autism research, although they are quite popular in studies of schizophrenia and bipolar disorder. So people have taken advantage of the fact that nowadays it's possible to create mice in which one can engineer a change in a specific gene. One can introduce a mutation in the gene or silence the gene or increase the number of copies of the gene. And so there are now a whole host of so-called animal models of autism, many of them based on changes in the genes that influence synaptic function. And so that's an interesting approach to trying to understand at a cellular level, in a way it's not possible in humans, what the consequences of changes in an individual gene might be. So in terms of where genetic research is going next, there's still concern that most of the genetic variation, sometimes referred to as heritability, has not been accounted for. So people are trying to identify bigger samples of individuals. At the moment, worldwide, there are only 2,000 or 3,000 individuals whose genomes are available for study. There's now the capacity to sequence the entire coding section of a gene, referred to exons, and in Canada, Steve Shearer's group has been doing that. But also there's increasing interest in looking at the genetic material in between the coding regions, and we refer to this section as introns, because this is where the control machinery for turning genes on and off is localized. And it seems quite plausible that for a developmental disorder, the abnormality may not be so much in the structure of the protein as the amount that is produced and whether it's produced at the right time in development. And so I think we're likely to see people shifting some of their attention to control of genetic programming as opposed to the nature of the protein itself. So I'm going to switch now very briefly to the neuropathology of autism. This is a very old slide just to illustrate that in one particular series, the weight and size of the brain in adults with autism was increased. So you can see here we have a number of very large brains. So in the typical population, normal brain weight would be about 1.45 to 1.55 kilograms. And so this sort of finding of an increase in brain size has now been replicated using MRI studies. There seems to be an acceleration of growth in probably starting about the second or third year of life. And then some disagreement in the literature about what happens next. So many researchers are claiming that autism involves an acceleration of growth and the rest of the population catches up later in development, whereas there are some studies suggesting that in adult life one can still find some individuals with increased brain size. And here we have a problem that relates to who takes part in MRI studies and what sort of individuals end up prematurely deceased and their material becoming available for post-mortem studies. So by and large, most post-mortem studies to date, and there are a very small number of these, and this is a bottleneck in autism research, tend to contain individuals who had both comorbid mental retardation and comorbid epilepsy. And those are not necessarily the sorts of individuals who take part in MRI studies. So it's possible that the findings both from the post-mortem and MRI studies are correct. It's simply that they're describing rather different ends of the severity spectrum in autism. Most post-mortem studies have found abnormalities in cell migration that occur prenatally, so this is a very striking example. This is the white matter here, and these are bands of neurons that didn't complete their migration to the cortex. So this is a very severe developmental abnormality, as is this. So here we have the cortical layering, if you follow the arrow, which has this sort of sawtooth pattern, whereas in the typically developing population this would be looking like a neatly made bed with the layers going straight across the screen. So this is due to abnormalities in the position that cells end up in as they may migrate from the ventricular epithelium up here at the top of the screen towards the cortical surface down here. And here we have the cortical surface, and this layer of the brain is referred to as layer 1, is normally pretty free of neurons, and multiple post-mortem studies have found big neurons in this layer that shouldn't be there. So these are neurons that have migrated too far. And in this particular instance, one can see that there is a cluster here of pyramidal neurons that should have been almost off the screen here. So whatever the problem is in cell migration, it's not simple in the sense of either not far enough or too far. It's more complicated than that. And what most post-mortem studies have found is that these abnormalities are patchy. In other words, if we move to a section to the left or right of the screen, we might see an area of cortex where there were no migrational abnormalities that were visible to the eye down the microscope. Another way of looking at the cortex is rather than making qualitative judgments, is to count cells and work out their density in different parts of the cortex. And this sort of approach gave rise to the mini-column hypothesis of autism that's been put forward by Manny Casanova. And the suggestion here is that the columns that make up the cortex, these are vertically orientated columns, are smaller and more numerous in autism. So in a sense, the pathology is in a horizontal plane. These are some data from a different group of individuals where now, rather than counting horizontally, we're counting within the vertical structure of the column. graphs at the bottom show autistic brain, and the two at the top show brain from typically, the two graphs on the left are control brains, and the two on the right are autistic brains. And if you look on the left at the moment, you can see there is a particular pattern of neuronal density as one goes from layer 2 of the cortex, where cells start, down to layer 6, which lies just above the white matter. And you can also see there's a relationship in both of these brain regions between the number of glia and the number of neurons. And what one sees in autism is firstly that the distribution of neurons isn't quite as isn't identical to the corresponding region in the control brain, but perhaps even more interestingly, there's a loss of the relationship between the number of glia and neurons. So if we take this with Manny Casanova's data, we're really looking at quite a complex, or potentially a complex, abnormality in the structure of the cortex that isn't definable by an abnormality in just one direction. Now, if cells end up in the wrong place in the brain, then a consequence of that is that they then tend to connect to the wrong cells in other parts of the brain, and this is a macroscopic example of this phenomenon. So this is a section through the pons of a child, which is in the brainstem. These are all tracks going from the cortex to the spinal cord. If we look at all our pons, we would see all of these tracks in all of us, but none of us would have this particular pair of tracks, which had never been identified before. The implication of this is not that this is the cause of the child's autism, it's simply an example of abnormal connectivity at a level that one can actually visualize. And in the same individual, these are abnormal tracks in the cerebellum. And in another individual, this is the caudate nucleus. These darkly staining areas are axonal tracks, but typically they'd be going at right angles to this direction. So they would look on the screen as though we'd thrown down a handful of pencils. So we have some suggestion from post-mortem data of abnormal connectivity. And you would think, well, why can't we use post-mortem material just to trace out abnormal connectivity? Because in animals, if the brain is removed quickly after death, it's possible to either inject tracers into the brain or put dyes on the surface of the cortex, and these dyes follow connectivity patterns, but sadly we're not able to do that in humans, so that there is a limit to what we can infer from post-mortem data about connectivity. Now, I mentioned earlier that tuberous sclerosis is a single gene cause of autism. It only accounts for 1 to 2% of people with autism, but approximately half of all people with tuberous sclerosis develop autism. And historically, the understanding was that this related to the location of tubers in the brain and the comorbidity with epilepsy. But over the last few years, people have been studying tuberous sclerosis more carefully and have realized that the disorder also involves abnormalities at the neuronal level. So some neurons are both too large and abnormal in shape, and some involve ectopic axons. And this opens up the possibility that tuberous sclerosis may be a better model for autism in general than we'd suppose, because it's possible that these microscopic abnormalities, which are closer to what we see studies of idiopathic autism might have a role to play in the development of autism as opposed to the physical location of tumors. We've developed many more cognitive models of autism since the 60s and 70s, when it was simply language versus ill-defined social abnormalities. And nowadays, most cognitive psychologists have moved away from the idea that autism is caused by an abnormality in just one of these areas, which has downstream consequences on everything else, to thinking that autism really involves abnormalities in many of these different processes. But these are such diverse processes and involving so many different areas of the brain, it's hard to argue backwards from psychopathology to what the underlying brain systems might be. And instead, people have tended to concentrate in imaging studies on finding differences in an individual system between people with ASD and typically developing individuals. And so imaging studies have really been investigating these sorts of abnormalities. So I've already mentioned increase in brain size. There have been a number of studies over the last couple of years suggesting that the gyral pattern of the brain is subtly different in ASD, which is of course what you might expect if there are abnormalities in cell migration. And indeed, some studies have suggested that if one examines MRI scans of individuals with ASD and compares those with scans from typically developing people, that you could use MRI to make a diagnosis of ASD. I don't think there's much future at the moment in that sort of path, but it is interesting to know that there are macroscopic abnormalities. People have also been interested in the idea that autism might involve an abnormality in long-distance connectivity. And so the thinking here is that long-distance connectivity is important for connectivity, for instance, between the functional lobes and sensory processing areas. And so one simple version of this account is that autism involves decreased long-distance connectivity and increased local connectivity. So this is connectivity at an anatomical level, and I'll show you in a moment how one can measure that. But another way of thinking about connectivity is functional connectivity. In other words, if one gives people tasks to do and examines their brain in some sort of functional imaging methodology, can you see abnormalities in a functional network? Or is there abnormal localization of function? Other sorts of system-level abnormalities that people have suggested have been abnormal bindings. So by that, I'm referring to the phenomena where different parts of the brain that might be involved in a common cognitive task seem to oscillate in synchrony with each other, and one hypothesis has been that this synchronous activity is disrupted in some way. Another hypothesis that isn't on this slide is the idea that autism involves an imbalance in the ratio between excitatory and inhibitory activity. Although these are quite separate hypotheses, I think it must be obvious to everyone that there's quite some overlap between these different types of account. So differences in excitation and inhibition, if they exist, are likely to reflect differences in underlying numbers of excitatory and inhibitory cells, and that might have knock-on effects on connectivity and possibly also localization of function. So the next few slides are really just to illustrate some of this point. So this is a study of theory of mind that we conducted in very able individuals in Oxford. Sarah Carrington did this for her PhD. And it turns out that in typically developing people, when we're engaged in theory of mind tasks, we activate an area called the precuneus around here in most theory of mind tasks. But when we're engaged in false belief tasks, we also activate an area here anterior to the motor strip. But in individuals with ASD, they're activating an area much more anterior. Now, using a different type of approach, this is data gathered from magnetoencephalography, or MEG, and one of these blue shapes is a representation of detectors over the head that started as a helmet shape and have just been flattened out. So this is the front of the head, this is the back of the head, this would be where the left ear is, and this is where the right ear is. And this is a false color map that relates to activity at this point in time when people are seeing different sorts of images. And so this particular figure is showing what happens in typically developing people when we see pictures of faces. And this area here is localized over fusiform gyrus, which is the face processing area in typically developing individuals. What we see in able adults with autism is firstly that the response to faces is less differentiated from other categories. The response to other categories of object, the response is weaker overall. But to a first approximation, it looks as though it is coming from fusiform gyrus. In fact, when one looks at the data more closely, it turns out that the localization in typically developing people is very similar across individuals. In other words, we're all tending to use a rather similar part of fusiform gyrus to process faces, whereas this area, in fact, is the culmination of individuals with autism activating their own little private patch of cortex around the face area in fusiform gyrus. In other words, each individual has a unique area of the cortex that they're using to process faces. And so that really begs the question of why this activity has ended up in slightly different places in individuals with autism. It also turns out that in typically developing people, we have some fast pathways in the brain that are activated before visual cortex is activated. So if you look at the top row of data here, you'll see this is very soon after an image is presented here at 60 milliseconds. And this looks like activity over fusiform gyrus that one's only seeing to the first face in a pair of faces. And although we have some fast activity when we see pictures of mugs, it's not at all over fusiform gyrus. What we see in ASD is actually there is no fast activity when in people with ASD when they see the first face in a pair of faces. And they do show some fast activity when they see the second face in a pair, but you can see it's akin to this anterior activity in typically developing people, and they're showing no activity over fusiform gyrus. So further evidence that the mechanism that people are using to process faces seems to be rather different from that in typically developing people. And the situation is even more complex when one carries out similar studies of children, because it transpires that typically developing children are activating fusiform gyrus very weakly before adolescence, and most of their face-specific activity is occurring in midline posterior occipital cortex, and that is true also of people with ASD. So what those sorts of findings highlight is that if we confine our studies just to adults, which often we do because of issues with cooperation or the nature of the task, we're seeing end-stage pathology, but that doesn't really explain what was happening earlier in development, and the differences in localization of faces are really an extreme example of that shift in localization. And this is just to take an entirely different scenario. Again, MEG data, this is now a reading task, and if you focus on the lower set of images, here these are typically developing people responding to the presentation of an incongruous final word at the end of a sentence. And what one is seeing is predominantly left-sided activity in anterior language processing areas, whereas in people with ASD we're seeing predominantly right-sided activity. We've subsequently replicated that study in a completely different set of individuals. And if you just look at the right-hand panel here, these lower traces are from individuals with ASD and no language delay. These peaks here are associated with processing of the visual word form, and you can see that in individuals with language delay, there is some delay in this activity, and it's also greater. But quite unexpectedly, it turned out that the right-sided activity that I showed you in the previous slide is driven completely by people with language delay. So in other words, individuals with autism and language delay are those individuals that seem to be using the right side of the brain more than the left to process language, whereas individuals with a more typical Asperger picture are showing the usual left hemisphere pattern of activity. Quite why that should be is currently unknown, but there have been a couple of studies recently looking at excitatory inhibitory balance in ASD measured using TMS, transcranial magnetic stimulation. And what was found was that decreased inhibition was only seen in individuals with a history of language delay. So this is an area that we're personally following up at the moment, because if we can identify at least one subgroup of autism where we can find a robust and replicable abnormality, we might be able to chase down the underpinnings of that. I'm coming up to the last couple of slides. This one is showing what DTI images of the brain look like. So DTI stands for diffusion tensor imaging, and this is a way of looking at white matter connectivity in the brain, as popular in neuroscience recently, and it relies on differences in the direction of water diffusion along axons. In other words, the water molecule finds it easiest to diffuse along the direction of an axon as opposed from side to side in an axon. And one can map out patterns of diffusivity. There have now been about 80 studies using DTI in autism. Just over half of those studies found evidence of a change in the measure of connectivity pointing in one direction, and then the remaining studies find either a change in the opposite direction or changes in both directions. So this is a very nice example of how neuroscience findings in autism tend to turn out to be very heterogeneous, and that might well relate to differences in the genetic risk factors that underpin autism in different individuals. And so it turns out that heterogeneity is a major issue in autism at every level of analysis. And I think what we can take away from that is that rather than thinking of autism as a localized abnormality in a particular system in the brain, that it's probably better conceptualized as dysfunctional networks as opposed to one key network, and that one can arrive at network dysfunction via a variety of different routes. It also seems plausible that some of the processes that we believe are intact in autism may be intact at a performance level, but may be subserved by rather different networks than we see in typically developing individuals. And this is the huge advantage of imaging approaches. It is an advance on the neuropsychological approach of looking for disjunctions in between preserved and impaired processes, to being able to look at the underlying neurobiology of cognition, where we might well find out that what appears to be intact from a neuropsychological perspective is in fact being subserved by quite different mechanisms. So I think my final conclusion is, at least at the moment, we have no dramatic breakthroughs on the horizon. This appears to be the case, as in much of science, of just continuing careful patient work and perhaps trying to focus particularly on subgroups of individuals with autism where we believe we've got a network that we can investigate, or a particular type of genetic abnormality that we can